introduction, and good afternoon. Friday afternoon. If I fall asleep, Ryan's going to take over. First of all, I'd like to thank Circa for hosting us and for Aries for hosting us this morning, and International Park Biotech as well. Visiting this area in Los Baños was really beautiful coming in, and I've been very lucky to see a little bit of the Philippines this week, and it's made me eager to look for opportunities maybe to come back at some point and do a bit more. But thank you very much for hosting me and for sharing with me what you're doing here. It's amazing. It really is world class, and you should be proud of the opportunities that present themselves here. I'm going to take you on a little bit of a journey today to talk about what I see as some of the global trends in biotechnology. If you were paying attention to the videos, you would have seen some of the things that have been happening over the last 20 years. Farmers' lives being transformed through this technology, biotechnology. And there are some new things happening in terms like gene editing and regulation. And hopefully we can navigate some of that through my talk and have you thinking about what biotech might mean for the Philippines going forward. But before I start, Rautaki Solutions is the name of my consulting business. As you would have heard in my bio, the word strategic and strategy came up a few times. That is what Rautaki means in Māori. It's a Māori word from New Zealand that means to be strategic or strategy. And that's something I'm very passionate about, is applying strategies to navigate regulation and to look at the pathway to market for biotech products. In this slide here, I'm just highlighting some of my clients over the last 9 years. Some of the logos you will recognise, some of them you won't. But nevertheless, in every case, each of these organisations are involved in biotechnology, and I've been lucky enough to in some shape or form assist them as they try to navigate regulation and the path to market for their biotechnology products. The very first global trend that I want to talk about is actually the ability to very cheaply and very accurately sequence whole genomes. So many of you will be aware that the human genome was sequenced quite some time ago now, and it's actually quite cheap, quite easy to sequence whole genomes. And in the context of agriculture, this is very, very powerful, because some of the genomes of plants and animals are really complex. Maybe not so much in animals, but certainly in plants, incredibly complex. The ability to sequence those is an incredible, powerful tool for our plant breeders and our animal breeders. And particularly now, because instead of looking at markers in a small subset of markers in the DNA and looking at their inheritance, we can look at every single base pair and treat those as markers. Very, very powerful tool, and I'll demonstrate some of that application shortly. The other innovation and the powerful tool that we are seeing, particularly in Australia now, is that farmers and our researchers and our breeders all have drones. And to those drones, they're attaching cameras, and they're using those cameras to capture images of their crops as they're being developed. So in this slide, you can see some small plots, aerial views of some of these small plots, and through the use of the imaging, we're able to very accurately take measurements of those plants. So whereas some of you that are students would have been asked to measure and weigh things by hand, we can now get that same data using imagery and drones at whole paddock scale level. So you can imagine how long it takes to measure an individual plant, the number of leaves, the width of the leaves, the length of each one, counting them, then looking at roots, then weighing the plant. Right? Even just saying that for one plant, imagine if you need to do 10,000 or 100,000. It's a very long, laborious process. We can now get that same information using cameras. Not only just the sort of camera that you have on your phone, but we can also attach cameras that look at different wavelengths, hyperspectral cameras. And the great thing about these is they give us insights into what the plant's doing. So we can use, for example, near-infrared cameras and look at moisture stress, water stress in plants before we can even see it visually. Very, very powerful way of being able to characterise plants at high throughput and large scale. We can even attach these things to tractors into buggies and run them over our paddocks rather than flying over them. I'm not so sure this would work in a rice paddy as much, but maybe we could start attaching them to the caravel. Imagine that, having some cameras on those as they walk through the paddocks. So what happens if you combine the power of genomics with the power of what we're calling here phenomics, phenotypic characterization? Well, there is an absolute innovation, a biotech innovation in plant and animal breeding that is being implemented globally at the moment, and that's called genomic selection or genome-wide selection. Now, the way this works is that it's taking all of those measurements, all of that data, and all of the genetics from what we call a training population. And that's essentially a selection of material that is incredibly diverse and from a range of different environments. And using that information and mathematics, creating an algorithm to assign a breeding value for a particular trait of interest. So, for example, a breeding value for yield, a breeding value for disease resistance, oil content, those sorts of things. And so instead now, as a breeder, once you have that information, instead of when you now do your crosses, instead of waiting for those plants to grow to maturity and assessing them for yield or disease resistance, you can take a little bit of their DNA in an untested population and based on the breeding value, predict what the yield will be, predict what the oil content will be. Very, very, very powerful. By way of example, in the animal production, in Australia, every single dairy cow has its DNA tested. And we have characterised, since back to the 1950s and before, or phenotyped our animals. We know the milking values and the milking rates of every dairy cow. We know the temperament. We know their size and shape. And using the same principles of looking at the DNA and looking at the characteristics and developing the breeding values, we can take a hair sample from a baby cow and with very high accuracy predict what the milking value, the milk production of that cow will be well before it reaches maturity. So when you are breeding your new cows for production, you don't need to wait and grow them for a long period of time before they become milkers. You can predict very, very early and select those ones that are high milkers. Very, very, very powerful. Every single breeding program has either implemented the strategy or is looking to implement the strategy. And the next version of this is to not just assign a breeding value, but an economic return value. So the forestry industry, for example, is looking at implementing this and assigning an economic value. So in forestry, I can plant my plant today, but it's going to be 20 years before I harvest it. So I need to know what the value of it is today and predict what the value will be in 20 years' time. So these are some new innovations, biotech innovations, that are transforming the way we develop new varieties of new breeds. Now, many of you will have had a cold at some point in time, yeah? You've all had a sneeze and a cold and felt ill. We know that that's from viruses, right? We know that there are things that make us unwell. We also know that there are insects that can pass some of these diseases and some of these health risks between us and between animals, such as mosquito-moving dengue. We also know that there are plants that are invasive weeds. If we get them into our forests or into some of our production areas, they take over and we have to try to get rid of them. They're invasive. We also know that there are plants that can cause us harm, either through thorns and spines or giving us a rash. And more importantly, we also know that there are animals that can cause us harm. These are all biologicals, biological hazards. They have the potential to cause harm to us as people and harm to the environment. So these biological hazards require a level of biosafety, an ability to implement some form of control around the risk. So how do we control the risk of a snake bite? We don't stand on snakes, right? Then, about 20 years ago, genetically modified organisms came about, where we were able to take genes from another species and put them into a production crop. We didn't know, and we were unsure and uncertain about what was the potential harm to us as humans or harm to the environment. Therefore, they were considered biological hazards. And as such, we needed to apply the principles of biosafety to assess them for their potential impact on us as humans and the potential impact of them on the environment. So this term biosafety has really been a very big part in the way that biotechnology has been implemented over the last 20 years. So you're all comfortable now that you understand what a biological hazard is and what biosafety is? Do you know what a GMO is? A genetically modified organism. If you're not sure, it's fine. Basically, it's a very broad term that refers to any organism where their genetic material has been altered using a process we call gene technology. And that gene technology process is actually quite broad. It involves a whole suite of different applications. And it applies to any organism, plants, insects, animals, microorganisms, fish. So it's a very broad term. And in Australia, this term gene technology, our legislation is called the Gene Technology Act, and therefore it is very broad and covers a wide range of potential biological hazards. Now, over the last 20-something years, the commercial production of GM crops is increasing, and importantly, the Philippines has provided leadership in information around what's been grown, where it's been grown, who's growing it, and what are the benefits, the realised benefits from these products. And the authors of these documents are sitting in the room, I say. And last month, they launched the 54th brief on the global status of commercialised biotech crops. This is an incredibly powerful resource showing us a lot of information around these potential biohazards that we've had. And if we look at the last 23 years, some of you in the room, I don't even think of 23. So before you were born, we were growing GMO crops. And in fact, since 1996, just over 2.5 billion hectares of area has been planted to GMOs. To put that into context, that's about the size of Denmark. I can't quite visualise that in my mind, but Denmark sounds like a pretty big country. And last year, again, nearly 200 million hectares of GMOs over 23 years. And these are the four main crops that have been planted, and you can see the areas. Large amounts of soybean, maize, cotton, and canola. And this information is gathered and disseminated by us here in the Philippines. Incredibly powerful bit of information. What you can see here, though, is that these crops have been around a while, and we've grown a heck of a lot of them. So you have to ask the question, if these are biological hazards, why are we growing them? If we dive into the ICA report, we can see information on the socio-economic benefits of growing these. The benefit to farmers, and you would have seen some of that in the videos prior to this talk. The improved quality of life for smallholder farmers. The environmental benefits in terms of reducing greenhouse gas emissions, improving soil health, the way in which farmers farm, not having to disturb the soil as much, conserving and providing greater levels of sustainability to the farm. And food security, because of the reduction in pests and disease, increased yields and yield security, and particularly through variable climates. This information is looked at every year, and it's there for everybody to see. And it comes from the Philippines. So where do you sit in the global scene, you're number 13. And here I am on Friday the 13th, and we're going to talk about Philippines number 13. If the lights and power go out, it's not my fault. What you can see in this table from the IAAA report is that the USA produces 75 million hectares of crops last year, GMO crops. 10 times the whole Philippines production area. 10 times your entire cropping area. You sit down at number 13, that's 0.6 million hectares of BT maize. It's about 10% of your cropping area. You can see that Brazil and Argentina, South American countries, grow a lot of GMOs. And where I am living at the moment in Australia, we're just ahead of you at 0.8 million hectares. And the only reason that we've slightly gone ahead of you is that we're now planting more GM canola than we ever have before. And in fact, for next year, every growing region, major grain production area in Australia will grow GM canola. You can also see that there are a range of other countries like Vietnam, Honduras, Chile, Portugal, and some Indonesia, and some African countries as well, all growing GM crops. So despite these big guys up here, there are still a lot of other countries that are growing GMOs, and you can see what they're growing. And aside from those 4 crops that we saw, and I'll talk a bit about why it's only those 4 crops thus far, we are now starting to see, and if you look at the ISA report, that there are new GM products coming to market that not only benefit growers, but we're starting to see some that benefit you and me as a consumer. We're starting to see, for example, products that are designed to have healthy oils. Okay, put up your hand if you've never heard of golden rice. If you haven't heard of golden rice, you shouldn't be in this room. Again, something being developed right here. You're in the heart of golden rice country, right? This is an incredibly powerful product that can save lives. And not only through alleviating vitamin A deficiency, you guys are now working on further enriching it to make more healthy rice by increasing not just the concentration or the content of nutrients like iron and zinc, but making it more available so that when we eat these products, we get the best benefit. And those sorts of biofortification then apply to other grains. These are very close to commercialisation. And the Philippines is at the forefront of some of these technologies. But we're also might be seeing in the media that there are also other opportunities coming along. We're hearing about CRISPR. How many of you have heard about CRISPR? You heard about it? You don't know what it is? I'll help with that. And we're hearing about it being applied to things like mushrooms and now animals, livestock production. Did you notice that in that ISA report that there are already 4 commercial crops? There weren't any animals on that list, right? We don't have any commercial GM animals. So this concept of gene editing is a fairly new concept that's been around only for a few years, although the technology's been around a while, and we're starting to hear about potential products through this gene editing. It's a gene technology. So therefore, it could have the potential to be a biohazard, couldn't it? Do we know what the effect of this is on our health? Do we know what the effect of these will be on the environment? Let's explore that a little bit. So first of all, what is gene editing? Well, there will be scientists in the room that probably can explain it in much greater detail than me, but I'm going to keep it really, really simple because I like to be able to explain it to my mum. Hi, Mum. Gene editing refers to biotechnologies that allow small and precise changes to the DNA. Small and precise. And we hear this term gene editing, but we also hear about a whole suite of other names. And I've pinched this slide from a colleague in Costa Rica who puts this up, and then he reads them out. I'm not going to read them out, but you can see that this technology is known by a whole range of different names. They're all examples of manipulating DNA, but it's different from a GMO. And unfortunately, as is the case with a lot of science, it's pretty complex to explain it to people. But I have a very simple way of explaining gene editing. And I know that some of the people that I've spoken to today also have simple ways to describe these technologies. And as you go away from here, I want you to think about how you might explain it to them. Okay. But put simply, gene editing is about identifying and somewhere in the DNA to make a very precise cut. And in that cut, we basically break the two strands of DNA. Okay. Does that make sense? We identify somewhere very specifically in the genome and we make a cut. That's essentially what gene editing is about. And there are a range of ways to do that. We can use CRISPR-Cas, we can use talons, we can use fingers, there's OD. There's all these other different terms or technologies that do the same thing, identifying somewhere very specific and making a cut. Now when we've made that cut, there are three possible pathways. The first pathway, every one of you in the room is doing right now. Your DNA is being broken up and repaired by itself. Right? So what we can do is we can make the cut and we can let the cell repair itself. And sometimes when it repairs itself, it makes a change. That's why I've got grey here. Lots of targeted mutation. I'm getting old. We just allow the cell to repair itself. And sometimes it makes a repair, a mutation that we're after. And that leads to a particular phenotype. The other way is that rather than letting the cell decide how it's going to repair, I want to tell it how to repair. So I'm going to provide it with a set of instructions, a template. And that template is going to say, I want you to change and repair this way. Does that make sense? And then the third option is that once I've made that specific cut, I put a whole new gene. And that whole gene could come from anywhere, right? That's a GMO, as we refer to them today. But what about these ones? Are these GMOs? Let's think about that. So the first thing we need to do is work out where do we make a specific cut? How do we make decisions about where we want to do these edits? It comes back to the power of genomics and genome sequencing. And because we can genome sequence and we can use the power of genomics and genomic tools, we're now better placed to identify where to make precise cuts and to what we want to make changes to be. So genomic programs are incredibly powerful and important. So how do we apply it? Here's one example of an application of gene editing in human health. Now, if any of you have studied biomedicine or read papers on biomedical research, you'll know that quite often they use the mouse as a model system for studying diseases and cancers. It's a mammal, right? So some of the gene mechanisms in the mouse is the same as us. And we can make mutations to genes in mice that lead to disease or cancers and study them. And many universities have animal facilities with lots of mice where they study diseases and cancers. But part of the problem with the mouse is that we don't look like mice. Well, some people do. But I don't think I look like a mouse. And I know I'm bigger than a mouse. So sometimes the disease that we see in the mouse is kind of like what we have, but it's not the same. And the other thing is that these things are quite small. Their organs in particular are very small. So how do I study things like kidneys and hearts and lungs in a mouse? It's not as easy. So what if we could use the power of gene editing to create those same mutations in a very targeted way in an animal like a pig? The pig at full size has the same size organs as us and similar etiology. So if we have a pig model, we're better placed to be able to develop new medical devices and new medicines. And in fact, here's an example. Polycystic kidney disease. This is the first model using the power of gene editing to specifically target a gene and develop a pig that has this disease. And as you can see here, that kidney is a lot bigger than the mouse's kidney. So therefore the researchers now have a better opportunity to study this disease and find potential cures and treatments. Sound good? I think it's pretty interesting. But we're in the heart of agricultural research. And I'm an agriculturalist, I love agriculture. So what are the applications of this technology in this space? I'm going to explore a couple of examples to give you some ideas on how the technology can be applied and for us to think about them as whether they really are biohazards. So firstly, I'm going to talk about herbicide tolerance and herbicide tolerant canola. This is a gene-edited crop that is now commercially available. It was developed in the US in San Diego by a company called Seabus. And it is resistant to up to 12 times the concentration field rate of a herbicide. 12 times. The mutation is a one base pair change in one gene. One base pair change in one gene means that this plant is now herbicide tolerant to 12 times the label rate. How interesting is that? And in some countries that would be considered a GMO and require heavy regulation. But I can make that same product in a number of other ways. How many of you here have done plant tissue culture? Put up your hand if you've done plant tissue culture. Plant tissue culture is a laboratory-based method of basically cloning plants. Right? And you can create many, many clones. In doing that process, sometimes we get mutations. Sometimes the DNA breaks and it repairs itself and creates a mutation. If you do enough tissue culture, you can identify and select for this mutation and get a herbicide tolerant canola. Right? But you may get a number of other mutations as well, not regulated. I can apply chemicals or radiation to that canola and force breaks in parts of the genome all over the place and hopefully hit this gene in that spot and select for herbicide tolerant canola. In fact, we have been growing herbicide tolerant canola that was generated through mutagenesis breeding using chemicals and radiation for more than 20, 30 years. Not regulated. Gene edited one base pair, and every regulator in the community gets a little bit upset and concerned. And concerned, for sure, is this a biohazard? It's got to be looked at in context. There are companies in the US and other parts of the world that are applying this gene technology approach, this break and repair, to generate a range of consumer-based products and farmer-based and consumer-based products. But for me, I'm actually really interested in what this offers for the livestock sector, and in particular animal welfare. I've always worked with plants. I've had a few students work on animals and it always made me feel a bit icky. I'm not real good with animals. Right? Animal welfare in Australia is a big concern. In fact, this is a taxi and this is a bus in Melbourne where they're trying to ban live export. This is a signal that the community is concerned about how we produce our food. In particular, things like live export or the removal of horns from our dairy animals. Did you know that we remove the horns from our dairy animals? Probably not. In the community here that we remove horns, they get quite concerned. But what if there are ways that we can tackle some of these welfare concerns through the power of gene editing? Maybe we can, maybe we can't. Remember, these are just tools that we have that may offer solutions to some of the problems. The first trait I want to talk to you about is heat stress. Now, the demand for milk protein and animal protein in Asia is ridiculous. The demand is massive. This is a huge opportunity for economies like Australia because we export a lot of the dairy products that we produce and we export meat. But we also export live animals so that you can be self-sufficient and have your own dairy herds. Here's the problem. A dairy cow that lives in Victoria, where I live, in the bottom of Australia, lives in air conditioning pretty much in an environment like this. What happens if you move it up here into the tropics? It gets a little bit heat stressed. If I go and stand outside, I come from a winter in Melbourne to 30 degrees and humidity, I get heat stressed. I sweat and my productivity goes down. Similarly for these animals, their milking values go down, their ability to reproduce goes down. They're very unhappy animals. Yeah. And that offers the question, why? Why are these guys like that? And yet 2/3 of the cattle population live in the tropical, subtropical regions. And those animals seem to be okay. So what if we go and look at some of these animals that seem quite happy standing out in the heat and ask the fundamental question, why are they different to a Holstein cow living in Melbourne? What makes them different? So we turn to our phenotyping and we turn to our genomics and we try to answer that question using science. And it turns out that those animals and the animals in Victoria have these genes called prolactin genes. We all have them. And it's like the thermostat. It's like your thermostat on whether you can tolerate increases in temperature. And by using the power of genomics, believe it or not, there's only a one base pair change in a prolactin gene that means that an animal will be more thermotolerant. One base pair. Do I have some tools to be able to create Holsteins that are thermotolerant? I do. So this is something that we're exploring, is to try to create high merit value animals that are good milkers with just one base pair change. What do you think the risk is to me drinking the milk from an animal that's had that one base pair change? These guys have it, and I've been eating them. You've been eating them for quite many hundred years, maybe 500 or 600. Interesting question. The other trait I want to talk about is the removal of horns. You saw that picture before with the animal with the blood, and that was a fairly old animal. Well, in the US, a company called the Combinetics created these two bulls here that were gene edited to have no horns. And the allele that was edited in there is the same allele that's in the beef cattle Angus. Angus cattle do not have horns. That Celtic allele we've been eating for 500 years. So what we did is put that allele in these Holsteins, and you can see they don't have horns. Now the problem is high merit dairy animals generally have horns. There are many beef breeds that don't. But a farmer needs to remove those horns. Needs to remove them for a number of reasons. Firstly, those horns can damage other animals. So when the farmer is herding in those dairy cows for milking, those horns might damage one of the other animals because they go through gates and have to go into the milking chutes, right? Those horns could also harm the farmer or the farmer's family, particularly if they're helping out with milking. And in addition, if we try to provide some health treatments to these animals, they need to be put into a contraption that basically holds their neck so that you can apply some drugs. Those that have got horns, you have to then manipulate the animal to get them into that position to be able to apply the treatments. So these horns are a problem for farmers, so they remove them. Okay. Now, the dehorning, or if you can imagine having your horns chopped off, probably not a very nice thing for an animal. And in fact, there is a cost to the farmer. In Australia, it's between $5 and $20 per animal. Right? $5 if I just chop them off, or up to $20 if I get a vet to come in, apply some pain relief, and burn the bloods when the animal is young. The problem is that quite often when you have large herds, you can't get to them at the young enough age, and you may not be able to afford to spend $20 on every animal. So what happens when an older animal has its horns removed, sometimes that animal doesn't make it. And in fact in Australia it's estimated that close to $140 million of product loss just from the removal of horns. And if you're like me, I get a little bit concerned about seeing animals with blood coming out of their head. I want to know that the animals are treated fairly and that they have a nice life. And generally the temperament of a dairy cow is pretty good once it's had its horns cut off and it's a few years old. So there's demand for dealing with this problem. So I said that there were two bulls developed in the US, and they've got no horns. And in Australia we imported some of that semen, and we did some, as a proof of concept, we put them into some cows. And because in Australia these are considered GMOs, they're in a certified facility and they're treated as GMOs. The way that they were generated was using that guided repair and instructions on how to make repair from a specific cut, to make them have the no horns exactly like the Angus beef. So we had 11 calves, and 4 of them we have confirmed that they are what we call homozygous for having no horns. And this is what they look like. You can see they don't have a horn bud, no horns on these girls, no horns on these boys. And the other interesting thing about animals that don't have horns, and particularly that same allele that Angus beef has, they have really long eyelashes, so they're really cute. Now we've done this in Australia not to commercialise them. We've done it to inform how we might go about creating a pathway to market for animals like this. Here is an animal welfare issue. We want to know, can we develop them within our regulatory framework and assess whether a product like this is a biohazard. And if you go to IRRI, you'll hear all about the quality assurance and stewardship program. A lot of that is about managing supply chains and ensuring that any potential risks to mixing and loss of the trade is managed. You have to do the work. It's an important part of being a producer of new varieties and breeds. So this is what we're doing in Australia. These are GM, and the next round of activity is to create thermotolerant dairy cows and thermotolerant beef. Have you heard of fall armyworm? This is a huge problem at the moment. It's become more of an issue in Asia more recently, but you've been a little bit lucky. Not completely, but a little bit lucky because you've got BTMOs, which is insect resistant. So to a certain extent, you've been saved by having the ability for your farmers to grow a GMO corn that has been assessed to be safe for human health and to the environment. And you would have seen in the video some of the benefits that those farmers are getting from growing these. Not just that, when these incursions happen, they've got yield security. Right? But there are some countries where they have banned GMOs, or they've had some huge amount of concern about biosafety, but now they're faced with an incursion like this. So the governments are having to make some very difficult decisions on ensuring they have access to all the tools and technologies available to them to deal with incursions like this. And we can see here, this is where fall armyworm, you can see in Africa, South America, and here through Asia. A real problem. So if you didn't have BT corn today, what do you think your government would do if you had this incursion? What would you, as citizens, want your government to do? Have you been reading the news? African swine fever. 100 million pigs in China already. I think some of the statistics that Ryan was talking about, that would be equivalent to a quarter of the pigs in the world that had to be put down because of this devastating disease. Now you remember the example I gave you around trying to work out how to make cows thermotolerant? How do we make pigs resistant to African swine fever? We've got some tools to look at this. We can look at genomics, we can look at phenotypes, and we can look at diversity and see where there's some natural resistance, and then see whether we have some tools to address this issue. So the Roslin Institute, based in Scotland, had that exact thought. And in fact, what they did is they found that this animal, this beautiful thing, the warthog, doesn't get this African swine fever. So what is it that makes this resistant and this one not? And through their research, they've identified this gene and 3 places where this gene differs between the warthog and the pig. So both animals have this gene, but this one has differences here, here, and here. Do you think we have some technologies to make this more like that in that one gene? We do. So that's their hypothesis. Perhaps through the application of specific guided repair of these 3 sites to make a pig more like a warthog in that one gene, have the effect of providing resistance to that African swine fever. That's what science does. We apply a hypothesis and now it needs to be tested. And through that testing, you're looking at not only whether these are resistant, but what is the effect of making those changes to these animals. That's what we do as breeders. We look at the effect of the changes in the crosses that we make. Does that sound like something that your government should be looking at? As a concerned citizen, are these the sorts of things that we should be looking at to solve some of these problems? These are questions I'm posing to you to think about. Those 2 things that I talked about are just the first of probably a number of challenges that we are going to face in agriculture, in being able to feed, clothe and house our people. And to satisfy the demands of countries that are becoming more affluent, the demand for protein in particular. So how do we deal with some of these issues at a government level? Well, in every case where we have GMOs being grown, there are regulations. There are rules on 1, assessing their safety as a biohazard, and 2, how they may be introduced into the food supply. And through that regulation, we have some differences around the world. And sometimes that can affect trade, and sometimes that can affect access to some of these products. So governments are trying to find ways to harmonise and work together to assess the biological hazard and ensure access where possible for these products. Regulatory cooperation is happening around the world and it's making a big impact, particularly in places like Africa, South America, and we're seeing even collaborations between Australia and Canada. And the ultimate level of cooperation is a mutual recognition agreement between Australia and New Zealand for food safety. Now, in every single country that grows or imports GMOs, as I said, 2.5 billion hectares over the last 23 years, they all follow a set of international guidelines for risk assessment and food safety assessment. There are rules and guidelines to assess the biological hazards, the uncertainty around these products. And what we find though is, see this number here, 370. This is the Roundup Ready gene. This is a gene that provides resistance to the herbicide Roundup. It has been assessed and approved 370 times. This is the BT gene that you have in your corn. It has more than 220 approvals from 30 different events. It's been assessed a lot, don't you think? And guess what? In every single case, here and here, not one differing opinion. The gene and the gene products pose no harm to us as humans, or to animals, or to the environment. In every single case. Now, let's assume Erie, or one of the other groups based here, have developed a new variety of something that happens to be a biotech product. There's a development phase, right? So development of a new rice variety takes about 12 years. Right? And it's probably not that much shorter even if it's a GM product in reality. But the research and development, the proof of concept, the testing of it, that's all within the control of the researchers and the developers. What's not in their control is gaining regulatory approval. So in order to get regulatory approval, you have to go through your regulator here for them to assess your product for human health and safety and protection of the environment. Makes sense? Potential biological hazards. But you don't just need to get approval here. If you're going to export this product, or you have other grains that get exported, then you need to get approvals in all of your other export countries. So you may need 10 or 12 or more approvals before you go to market. What do you think that does to the research and development time? It blows it out. And there's no control for the developer. There's a lot of uncertainty as to when all those approvals come in and when you may actually be able to recoup your investment. It's estimated the investment of taking one product, one variety to market is in excess of $130 million US. Does Erie have $130 million to commercialise one variety? I don't think so. We asked your Congress, members of Congress on Monday, if they had $130 million to assist in the commercialisation of a biotech product developed here in the Philippines. They don't have $130 million. So when you see only 4 crops dominating the number of hectares being grown, the reason it's only 4 is because 1, those products, the cost of developing those products can be recouped because they are broadacre crops that are grown extensively around the world, and so the value capture to recoup $130 million is there. You may not be able to recoup $130 million from a virus-resistant white clover or a virus-resistant pike. Okay. So, organisations like Erie and some other public sector organisations cannot commercialise biotech crops because regulation is a blocker. And as I showed you before, these products get assessed multiple times by multiple regulators using the same sets of international standards and guidelines, and in every case, the products are deemed safe. So, I would suggest that if you do the same thing over and over again, trying to expect a different result, it's crazy. I think there's going to be something a little bit about that. So how do we deal with this? Well, there is an opportunity because there is this redundancy, this duplication. So this is the number of food approvals, food safety decisions are made in Australia and New Zealand, here in South Africa. So these are the numbers, right? There's a heck of a lot of duplication, a duplication of effort. So because we use the same sets of guidelines and standards, maybe our governments could work together a bit better. This data is a few years old now. It's now more than 2,000 food safety decisions made on GMOs. And I'll repeat it again, in every single case, safe for human consumption, safe for animal feed, and safe for the environment. Not one different opinion across all of these different jurisdictions, including Europe. This is actually something, a global trend at the moment, is that governments are recognising this and trying to find ways to do it better. Now, coming back to those products we call gene edited, how do we tackle that from a biosafety, biohazard perspective? How many of you here are aware that your signatories to a Cartagena protocol for biosafety? Have you heard of that protocol? Maybe not. This is an international convention where a large number of countries have signed up to it, and it's about how to manage the potential risks of these products, and in particular looking at the movement of them between different countries. The powerful thing about the Cartagena protocol is that in it lies a definition of what a GMO is. In this case, it's called a living modified organism because it refers to things that are living, not dead. But importantly, the definition says this, that it really is something, an organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. The same definition in all those countries that have signed and ratified the Cartagena protocol, and there are a lot. So, what does that mean in terms of some of the products that I've talked to you about? Thermotolerance in cattle. 2/3 of the cattle that live in the tropic, subtropical region have that one base pair in their prolactin gene that makes them tolerant to that. You guys have been eating it for at least 500 years. Is that a novel trait? Is that novel? If I make a Holstein in Australia have that one base pair change, is it novel? I would argue that it's not. The genetic dehorning, the Celtic allele that's been in Angus, we've been eating beef, Angus beef for 500 years. You put that allele into a dairy cow, is it novel? I would argue that it's not. So if we apply this test, those animals, the biohazard, the potential biohazard to us as humans with the environment is in there, and therefore we perhaps should be regulated. So this definition and the consistency of this across members, those signatories to the Cartagena, offers a way to be able to make some decisions about whether we regulate and restrict these products coming to market, the opportunities that are there. Many jurisdictions are struggling to deal with GMOs and gene-edited new breeding technologies. And the reason why is because they're, in particular, not signatories to the Cartagena Protocol. The EU have said it's all bad. Very, very precautionary. The USA won't regulate gene-edited crops as long as they satisfy certain criteria. In Australia, we're a little bit half there and half not. I'm not talking about that. But the reason we're half there and half not is because we did not sign the Cartagena Protocol. We have a different definition. That definition is set in law, and that restricts our ability to be able to rapidly adjust our regulatory system. It's hard to change the law. South America is leading the way. South American countries and Central American countries are signatories and have ratified the Cartagena Protocol. They have the same definition for an LMO or GMO that you have. Here's the model. You have something that has been gene-edited. Let's take our thermotolerant cow. This one's got horns, I can't call this horns, right? So let's see, we go and talk to our regulator. We have an early consultation and we consult with the Biosecurity Committee. I reckon that's great. It doesn't matter whether it's a GMO or not, if it's a product of biotech, we should be talking to our regulators and having them ratify what we think. So let's have a consultation with them, and then we use the definition out of the Cartagena Protocol, which is there a combination, a new combination of genetic material? Those words are familiar? The answer is yes. It's a GMO and we regulate GMOs now. Potential biohazard, let's assess it. But if it's not, we have to ask another question. Does the breeding method use a transgene, a GMO, temporarily? The answer is no. Why would we regulate it? An example of that is making a specific cut and then the cell will repair itself. Does that sound like something, as long as it's not novel, that we shouldn't be regulating? I would agree with this model. But what if it does use a transgene temporarily? Then we have to ask another question. Does the final product have the transgene on it? If it's not free of the transgene, then it's a GMO and we should assess it. But if it is free of that, again, it's a break, but there's no template there. It's joined, but it's repaired the way we want it to repair it. And as long as it's not novel, we shouldn't need to regulate it. Very simple model that works and leverages off a very beautiful definition. Do you think this is a sort of model that the Philippines should be looking at in terms of how it makes its decisions on whether to regulate or not certain products from gene editing? I think this is quite a nice model. The reality is, pretty much all of South America and Central America have already adopted this model. They're already approving products using this decision tree. What will the Philippines do? What will ASEAN do? I can tell you what we're doing in Australia. We're stuck. We don't have your definition. We actually have two definitions. A definition from our Gene Technology Regulator, from our Gene Technology Act, and one from our Food Authority. So we've been trying to review, to try to find a way in which we can assess the potential risks of these products and how we may or may not regulate them. Our regulator undertook a technical review, and the outcome of that technical review is that allowing the cell to repair itself from a specific cup may not be considered a GMO come October. Yay. However, if I want to tell the cell how to repair, and I guide the repair, unfortunately it will be considered a GMO, regardless of whether it's novel or not. What that means is that we have to somehow try and change our policy setting, which is a change of our legislation. We reviewed our legislation. We had 27 recommendations, one of which was to look at it. We only review our legislation every 5 years. How far behind South America are we now? Australia's 5 to 10 years behind them in how we deal with some of these opportunities. And to make things more complicated, our Food Authority is also looking at this, but they haven't made a decision yet. If they don't make a decision by October, what it means is that we may have a situation where I can take a product to my Gene Technology Regulator, that consultation phase, and they can tell me, your product's not a GMO. We're not going to regulate you. But if it's a food, I have to go to this authority, and they're going to go, well, under our current definition, it is a GMO. So we have domestic asynchrony in our regulations. What do you think that does to Australia's competitiveness? It's not great, mate. And if I'm trying to develop something in Australia, where's my pathway to market? My pathway to market is in South America. Or maybe it's here in the Philippines. This is just what we will have in our regulations. We will have a statement that says that if we use a template as a guide, it's a GMO. If we don't add a guide, it's not a GMO. So that's kind of what it is. So we will kind of have some opportunities in Australia, but then there will be a whole lot of other opportunities that we won't have. Not here. So, sort of to conclude my talk, what I want to do is just highlight a few things. First of all, the role of biotechnology in agriculture continues to increase. And if we look at the data that ISO have been providing, all but one of the years that we've been growing GMOs, we've had double-digit percentage increases in the area for GMOs. So I'll ask you this question. Why are we growing so many of them if we're bad? Look at the ISO report, you'll see why farmers are growing. Why we have 2.5 billion hectares over the last 20-something years. The other thing that's really important is we're starting to see products that are more centered around us as consumers, not just for the farmer. We've seen the benefits for farmers. I want to see what these benefits will be for us. Healthy oils, tackling issues like vitamin A deficiency or iron deficiency, zinc deficiency. Interesting. Hold your breath. The other really important thing is that it's not the big, large multinationals that you might think that are taking the next generation of products to market. It's a second tier of companies that have worked out ways to navigate regulation, where some of the pathways to market are opening up as regulatory systems start to cooperate more, and the price is starting to come down on some products. I think that's a growing trend. And if some of the opportunities from gene editing are realised through the not being considered GMOs, we will see a lot more biotech products on the market. Chile have already approved 3 gene edited products already now. These new technologies, though, do offer new challenges, and we have to, as the regulated community and as concerned citizens, need to consider those challenges. But we need to look at them case by case and look at the potential risks, and we need to be pragmatic about what that means. Some of these environmental impacts that we see, fall armyworm, African swine fever, we're only going to see more of these. And when they become issues of national significance, national importance, governments are going to have to make very rapid decisions. It is our responsibility as the consumers to empower our leaders to be able to have the tools to make those decisions, and that our producers have all the tools available to them to solve these problems. We have to think a bit more big picture. And in fact, what I'm talking about here is a product pull model rather than a tech push model. So here is a problem. What are the tools that I've got to solve this? Gene editing might be one, GMO might be one, it might be genomic selection. I've got a toolbox. But if I haven't got all the tools in the toolbox, if I'm missing the hammer and I've got some nails, I can't build the house. I want to be able to product pull rather than tech push. Regulation is important. It makes a big difference. It provides us with trust and certainty that the food that we're eating is safe and that there are going to be no impacts on our environment. We have to manage potential risks to us and to the environment. But we need to do that in a pragmatic, case-by-case, science-based way. If we can't bring emotion into it because we feel that it might be bad, let's use some science and trust our regulators using international standards on how we look for things like potential allergenicity, toxicity, etc. Let's look at what is happening in South America as an example. They're leading the way. They'll leave everybody behind. Stewardship of products is absolutely pivotal. We have to protect markets, trade, and particularly once we've developed stuff, we start importing and exporting. So learn from the stewardship program that's been developed for GMOs and have some trust in our industries that they have these stewardship protocols and practices in place already. Industry is already managing the segregation of different products. How do you segregate canola, barley and wheat through trade? We've been doing this for a long time. How do we segregate the different types of wheat, fig wheat and bread wheat and noodle wheat? Industry does this already, and they do it through stewardship programs. Let's have some faith that the system won't break because we bring a new product to market. New products come to the market all the time. But having said that, there is a balance that we have to have between managing potential risk and innovation. We need to be innovative, but we also need to be cognizant of the potential harm to us and to the environment. And with that, I want to thank you for your attention and let's have some discussion.